Hello, I'm Ava, and my project is on Pseudomonas aeruginosa vaccine development, or rather, a very small aspect of Pseudomonas aeruginosa vaccine development. I'll get into that later. But basically, at the beginning of the school year, we were supposed to be researching areas of science that are interesting to us, trying to find a topic that is interesting to us. And one thing that I have always been so interested in science is the immune system. So I kind of knew I wanted to look at something that was immune system related for my project. And while I was looking at different papers, like looking at different topics, I came across this topic. I came across a lab that is near me that was working on developing a vaccine against Pseudomonas aeruginosa. So I'm going to go into a little background of that right now. Basically, Pseudomonas aeruginosa is a bacteria that causes infections and often hospital patients. Uh, it's diseases such as pneumonia that can be caused by this bacteria. And treating these infections can be a bit tricky because the bacteria is very resistant to antibiotics. So naturally, people thought, well, antibiotics aren't working. We should try creating a vaccine against it. And there have been many attempts at creating a vaccine, but there have been no successful vaccines yet. So the lab that I was working in, Dr. Catherine Wall's lab, they created their own vaccine that they were hoping might work at preventing infections of Pseudomonas aeruginosa. But a vaccine needs to be tested, so they had groups of mice. They gave one group of mice this vaccine they created, which was created using the Pseudomonas aeruginosa protein and a new adjuvant they developed in the lab. And then a different group of mice as a control was given a vaccine that was created using the same protein, the same Pseudomonas aeruginosa protein, but this time the sigma adjuvant instead. And then another group was given the new adjuvant alone without the vaccine. So they had these different groups of mice, and then the mice were sacrificed, and their spleen cells were reintroduced to the Pseudomonas aeruginosa protein. And here's where I come in, because we wanted to see how are these immune cells in the spleen reacting to being reintroduced to this protein. Ideally, we would like these immune cells to be releasing something called interferon gamma. And the real important thing about interferon gamma here is that it promotes the production of the antibodies that fight against Pseudomonas aeruginosa. So we want a good, really strong interferon gamma reaction upon being reintroduced to the protein. So after my spleen cells were cultured with this protein, I was then given the supernatant fluid from the cultures to test the interferon gamma concentration. So the overall purpose of my part of the experiment was to measure and compare the immune response of the different groups of my spleen cells. But my specific question was, what is the interferon gamma concentration of these different samples? So to do that, I'm going to move my face over here so that you can see all of the methods here. But I did basically sandwich ELISA for measuring the interferon gamma content. And over here is a little drawing I did of how this ELISA works. So basically you've got your capture antibody that binds to the plate, and then if there's any interferon gamma in the sample, that will then bind to the capture antibody. After that, there's washing between each step, so anything that doesn't bind to something that's already there will be washed away in between each step. But the detection antibody next, if there's interferon gamma that is bound to the capture antibody, the detection antibody will then bind to that. And then the biotin that's attached to the capture antibody there then binds to streptavidin, which is part of the streptavidin HRP conjugate. And the HRP would then react to the substrate, which is the last step, is adding the substrate. And this would produce a color reaction. But if there's no interferon gamma in the sample, then the detection antibody has nothing to bind to, the streptovidin HRP conjugate has nothing to bind to. That all gets washed away in the wash step, and then there would be no color reaction if there was no interferon gamma there in the first place. So that's basically how an ELISA works. There is a bit more detail in my lab report if you want to look at that. I am going to move my face over here again. But this table here is showing for the standard what the average reading was. For individual data points, you can also look in the lab report, because this is just the summary table. But basically, this is used so we can see, because we're able to see what the different color reaction was, but we need to be able to compare that and put that to numbers for what is the concentration of interferon gamma. So we take these known, the standard has a known amount of interferon gamma, and which you can see along in this graph here, and then this is showing what the reading was. So this creates something called the standard curve, and this is what we compare the readings for the samples against. We just pay attention really to this part here that's linear. As you can see, it's real linear for a while, but then we have over here where it becomes not linear anymore. We don't really look in this area. Everything we look at is more in this linear area here. So then these were the average readings for the samples. Again, you can look for the more detailed data table. It's in my lab report. But basically, you can compare the readings from this table against the standard curve to get an idea of how much interferon gamma is there. For example, if you look at the vaccine 96-hour group, the reading was 0.03, and if you then compare that against the standard curve, that corresponds to a concentration of less than 0.1 nanogram per milliliter of interferon gamma. By comparison, if you look at the Sigma vaccine 96-hour group, which, remember, that was a group we were using kind of as a control to see how the new vaccine does compared to this vaccine that's just using the Sigma adjuvant. But if you look then at the value for that when going back and comparing it against the standard curve shows a reading of, shows an interferon gamma concentration of around 0.5 nanograms per milliliter. So it's 0.5 versus less than 0.1 for comparison there. And then over here I have just a graph that's showing the reading for each sample. Over here is the vaccine group, and as you can see, it isn't showing as much of as a reaction, because the higher, to put things simply, the higher the reaction here, the higher concentration of interferon gamma. And if you look in the control groups here, we have the Sigma vaccine and Cone A, which is just something that's used to stimulate the immune cells. It's used as another control group. But these groups were showing a much higher interferon gamma response than our vaccine group. So that unfortunately means that we are not getting the immune response we want out of this vaccine. We would like to see a much better interferon gamma response, because interferon gamma is needed to promote the production of the antibodies to fight against Pseudomonas aeruginosa. So the vaccine will need to be further developed to try and get a vaccine that can have a better interferon gamma response and hopefully be more successful, so it can eventually be used to prevent infections of Pseudomonas aeruginosa. And lastly, I just would like to say thank you to the wonderful scientists of the lab I was working in. It was Dr. Catherine Wall's lab. I would like to say thank you for allowing me to work there and take part in this project. And I would also like to say thank you to Dr. Shadia Nada for mentoring me throughout this research process. And then lastly, I have my references here. You can also see it in my lab report. Thank you for listening.